Good morning, good afternoon, good evening. Welcome to the Introduction to Forensic Science Week 3 Google Hangout, coming to you live from the University of Strathclyde in Glasgow. Today we'll be putting your questions to Professor Jim Fraser and Dr. Bill Tilston, discussing Week 3 of the course, Blood Pattern Analysis and DNA. Please feel free to ask questions using the Google Hangout question box, and we will do our best to pass along as many of your questions as we can. You can also post your questions on Twitter using the hashtag FLForensic. And now, without further ado, I will hand over to Professor Fraser and Dr. Tilston.
We've got some really interesting questions to deal with today. And the first one we want to talk about comes from Susan Bush. And Susan asks, is there any chance that someone can explain why mitochondrial DNA remains sort of unchanged over generations? And what does this mean exactly? Thanks. Well, we'll try, Susan. I think the first thing I have to say is I suspect that the way you phrased your question isn't exactly what you intended. Mitochondrial DNA doesn't remain unchanged over generations in the sense of when it's inherited through one generation to another one. It undergoes mutations exactly the same way that nuclear DNA does. And in fact, the mutation rate's higher in mitochondrial DNA than it is in nuclear DNA. I think what you were getting at was the thing we talked about in the module, which is there is so much more mitochondrial DNA in the body. It's roughly 100 times, or a minimum of 100 times more in a cell, because there are 100 or more mitochondria there compared to just the one nucleus. So that when we're looking at remains or debris from an aircraft crash or something in military, people killed in action, anything like that, or the Romanov case that we talked about, there's a lot of DNA there. So even though the material is old and degraded, there would be enough mitochondrial DNA there in good condition that we can in fact get a profile out of it. Jim?
Okay, thanks, Bill. Thanks, Bill. Good afternoon, everyone. This question comes from Matt Connolly, and Matt is interested to know if semen or material from a vasectomized man can produce a DNA profile. And Matt makes the point, of course, that a man who's been vasectomized ought not to be producing sperm, and suggests that it might be possible to obtain a profile from other cells in the seminal fluid. Let's just deal with some kind of basic issues here in terms of the particular body fluid we're talking about. Semen consists of two parts: seminal fluid, a kind of liquid component or a kind of gel-like component, and the sperm. And the vasectomization process prevents sperm from going into the seminal fluid. Cellular material, so from the individual who deposited the seminal fluid. In terms of finding that seminal fluid, you would use the same kind of tests to detect the body fluid, and then the DNA analysis. So that's for the establishing that the material is there. But as Matt suggests, or that he is aware of, that you can obtain a DNA profile from that seminal fluid, from epithelial cells and other cell types that are mixed in with the seminal fluid.
Thanks, Jim. Next question we want to deal with was posted by Rachel Hosier, and what I've actually said in introducing it is I'm going to paste the question I posed in DNA analysis part 1. That's about PCR process. Before I go to a question, I'd like to read the next part and respond to it. She actually says, I've enjoyed the challenge of this section, exclamation mark. And I think a lot of you have found this week with DNA very much more challenging than we did week 1 in crime scene or week 2 in fingerprints, and certainly than we will find next week on footwear marks and also week 5 and week 6. But to a certain extent, this was deliberate on our part. We don't want to turn you into DNA analysts or DNA experts. But DNA is such an important part of today's armoury in forensic science that we felt we had to give you at least information that you could delve into to the depth that you wished and the depth you were capable of, to get as much understanding as you could of this very, very important technique that is available to us in forensic science. We knew before we wrote the thing, and we knew from feedback from last year's class, that those of you who do not have a recent science qualification with molecular biology in it would find some of the stuff hard going. Just the names. We might as well try to teach you a foreign language in some cases. But what we've tried to do is keep emphasising, you don't have the background ability and you don't have the desire to get really into that degree, don't do it. Stay up there at the helicopter level. As long as you know what DNA can do for you, roughly how it works, and in particular that it's really, in a sense, replicating what the body does in a test tube, and instead of looking at it for the whole DNA, we're not talking about Jurassic Park here, but just looking at the wee bits of it that give us these STR markers that we can build up an idea of how common or uncommon is a type of semen recovered from a rape or blood recovered from a offence. That's what you want, that's fine. So that wee bit of background onto Rachel's actual question, which is, I think I understand how it works, and what my understanding is that you need primers, you need the Taq polymerase, you need free nucleotides, and after that, the question is, okay, so you need these, and the material told us a little bit about where the Taq polymerase comes from. It's from the hot springs, it's thermos aquaticus, that's what the Taq stands for, and it's important because the reaction that's used heats the material up, so this enzyme's got to be able to work at temperatures that are higher than body temperature for it to work. But nothing was said about where we get these other things from, and the very mundane answer to that is we buy them from chemical companies who manufacture them, wrap them up in kits, and sell them to the labs probably for a reasonably comfortable profit. So if you try and answer that, that's the way it is, Richard. Well, Jim's blocking out.
No,
the next question.
I know the next question. I wasn't sure if that was a pause for breath or a real pause.
All of the above.
I was going to just endorse one of Bill's comments there, really, in relation to DNA. Yes, it is quite tough going, but really think about this from a kind of criminal justice perspective. This is really important evidence that in many instances is involved in the conviction of individuals who may well be getting, quite heavy sanctions, including imprisonment. So it's important that as many people as possible have some grasp of how this process works, and it isn't just a kind of buzzword, or there's an automatic assumption that because you have DNA evidence that it's case closed. Shifting completely to another kind of very powerful evidence, this question comes from Sarah Horton. She talks about a case in Australia, which I don't know about the particular case, and the point Sarah makes is that in this case there was a fingerprint which appears to be a bloody fingerprint, or a fingerprint in blood, and this was described as a negative, rather, she doesn't say it's not positive, but a negative print. Now, Bill and I have discussed this, and we both come across this phenomenon, and I think the first part, or the main answer to Rachel's question, is that this is not amazingly uncommon. It's not common, but it's not certainly not unheard of. When you place a finger on a surface, if the skin is flexible, it's plastic, and it's curved, and when you place it on a plain surface, it would deform. And the more you push it into that surface, the more you will affect how the impression is deposited. And if you press it in very heavily, you will in fact reverse it, and you will leave an impression of the furrows. You will leave an impression of the furrows rather than the ridges of the fingerprint. And of course, that's easier to do if the fingerprint has some kind of medium like blood on it. that fills the furrows. So it is entirely possible, either by use of pressure or by having a medium on the skin like blood, to produce a negative mark. And one of the important judgments an expert would need to make when examining it was making a decision about whether that's a true positive mark, in which case the comparison will be between ridge to ridge, or any issues around having a compressed mark like that. By way of a reference to a case that, for those of you who are particularly interested in this, there was an important appeal case in England about 3 or 4 years ago where, and this was in a homicide where this was critical evidence, where the two experts for the prosecution and the defence could not agree whether the mark was a positive mark or a negative mark, which I think is really, really quite extraordinary. That there was such a big difference in their perceptions of what they could see from their observations. Bill?
The fellow I was going to use while Jim was catching his breath was, I think, an intentionally humorous question from Robert Knight. And Robert wants to know where Mrs. Ward's twin is. Well, I don't know, maybe we should chase that one up, Robert, but I think what you're really getting at there is giving us an opening to explore just a little bit more of this question about DNA in twins and how we would deal with it in reality. And of course, the actual fact is that if Mrs. Ward, let's say she wasn't deceased, but let's say that she was the accused, and she had a twin, and her defence says it wasn't me, it was my twin that did it, what can we do about it? And the answer is, from DNA, not very much. But as we keep coming back to, we still have these things, these nice things at the end of our fingers. And Mrs. Ward's twin's fingerprints will not be the same as hers. So if we're looking at trace material left at the scene, and that includes blood or sweat, DNA is not going to help us differentiate. But if it includes finger marks, then we're onto something that might in fact help us. That then leads me to the next question, which is from Robert Hart and Therese Murphy, both of whom have featured quite a bit in the discussions, particularly you, Therese. I've enjoyed your contributions very much, thank you. And they want to know about DNA fingerprints and DNA in the bullet casings, and fingerprints being identified in various places in all the samples taken at crime scene damage. We're not going to answer that here, because one of the things that we're trying to do is take you through in a measured rate the development of the case as it happens, and then deal each week with the aspects that are covered in the theoretical stuff behind it. So really good questions, and if you want to know the answer, tune in to the next exciting episode of Murdered by the Law next week.
You're going to pick up 13 as well?
13 as well, okay. That's from, oh yes, from Gail Williams.
Quality assurance.
Yeah. You wouldn't know, but Jim's having a wee dig at me here. Gail wants to know about, basically, the questions about quality assurance, which is an extremely important part of any forensic lab worth its salt. But if you thought DNA was full of funny words and boring, it's a thriller compared to QA. But basically, I'll pick up the questions and see if I can give you just a flavour of what's involved in this. Gail's first question is, the lab test equipment has to be calibrated and test certificates issued. What checks are in place to ensure compliance? What she means there is, particularly with DNA, this is all going through these things that we sort of slightly tongue-in-cheek call black boxes. We want to know that they're working. We want to know that they're giving the right answers. And the way that this is done is by calibration, putting known standards through them, reference materials through them, making sure that they're working properly, and then making sure that the people who use the equipment know that it's gone through those checks and that it's demonstrated working properly. And that's where the test certificates come from. And this is tied in with what's called accreditation. So basically, outside agencies come in with a set of standards that cover this sort of activity and run the ruler over the lab to make sure that it is meeting all the standards that are described there. For those of you with a wee bit of knowledge about it, the basic standards that are used in the lab is ISO 17025. If you don't know what that means, don't lose any sleep about it, it doesn't matter. If you do know about accreditation and ISO, that'll point in the right place. And there is, in fact, to assist the people that are coming in to do it, a quite lengthy guideline published by another organisation called ILAB, Inter-Laboratory Accreditation Cooperation, that gives, if you like, translations of how these different standards apply in forensic science. Gail's second question is, there are new technologies, equipment and practices that occur, so who's responsible and accountable for the latest technologies that they're following? Basically that's up to each lab. They can make the decisions about when they want to move on, when they don't want to move on, with the caveat that this in fact is one of the intriguing issues, particularly in relation to DNA and DNA databases. The first databases that were put in place had 4 STRs in them. We're now looking at databases that are accepting information from 20 or more. So somebody somewhere has got to make a decision that the evaluation of these new STR markers is sufficiently robust that it's legitimate to add them to the database. And then they've got to answer the question, well, what do we do about the old ones where there's not so many there? And the short answer to that is, you just have to accept that there's not as many there. Third question is implementation of new and best practices, who monitors it? And the answer to that is really a subset of the answers to the first QA question. Question 4, do any of the UK technicians carry out secondment to see how other countries carry out tests of similar requirements? This is a global issue here, that basically we like to pretend that science is a global community where we're all interested in interchange and exchange of knowledge. The fact of the matter is, practically every forensic lab in the world is part of government, and we all know that governments these days don't have an awful lot of money to spare. So yes, it does happen. It doesn't happen nearly as much as you would like, and it probably doesn't happen as much as would be ideal. And finally, our last question, if tests are carried out on an out-of-calibration equipment, would this sample be null and void and inadmissible? Well, if they were, it sure as heck should be null and void and inadmissible, but more importantly, and go right back to your first question, Gail, we would hope that that never happened, because one of the standards for the accreditation is that out-of-service equipment has got to be so labelled or taken out of the lab. So a very legitimate concern, but not one in practice that we shouldn't come up against. So there we are. 5-minute introduction to QA and CanLab.
And on that global point, it might be worth considering what is a kind of fundamental difference in forensic science from pretty much the rest of science, in that science is generally global. Science in Glasgow is the same as science in New York or science in Beijing, but forensic science in New York is not the same as Glasgow or in Beijing because they've got different legal systems. So these international standards are particularly important to try and establish standards that everyone can share so that material can be exchanged. And that takes me to the next questions. I'm going to deal with 2 questions here. Question 14, which is from Barbara Priestley, which is, if I read it out, with the number of segments of STRs on a strand of DNA, how does the lab choose which STRs to duplicate for the testing of the DNA during the crime investigation? And I want to link this to a question from Wendy Hall, which is about the loci. And Wendy says, does the core loci used for CODIS, that's the database in the United States, show any medical information such as a defective gene or precursors, for example, for breast cancer? I'm going to essentially link those 2 questions to address the kind of the broad issues that are being raised there. How does a lab choose, or how are the target loci chosen? They're chosen for a number of reasons. The features that an ideal loci would have for forensic purposes are not the same as an ideal loci for genetic disease screening purposes. The first and general comment I'll make is that to our knowledge at present, and there has been a great deal of testing, none of the loci are linked, nor are they specifically or clearly linked to any genetic disease or disorder that we know about. There is perhaps one that Bill and I spoke about that is tentatively potentially linked, but they've been quite rigorously tested, and it's important that they're independent and not linked because it affects the calculation of the probabilities in quite an important way. So they're not linked. They're not linked to any known disease state or any known genetic condition. What other features do they have that are important? Well, the critical one really, there's two perhaps very important ones in these choices, and the first one is that the material, the target site, the loci target site is capable of discriminating individuals within a population. It's the technical term for it, which I'm sure you'll have come across in the DNA module, is polymorphic. In other words, it could tell myself and Bill apart, or someone else, or another. The phrase that would be used is two people chosen at random in the population. Can they be discriminated? So the first thing, key issue is whether it can discriminate well people within a population. Then there are a whole range of other things that come after that. One of the key ones is that it's stable, and that the site doesn't undergo easy degradation. And when it comes to the actual choice nowadays, it's quite a complicated one because, well, it's heavily influenced perhaps by commercial interests, because pretty much everyone is using the same kits these days. That's one of the benefits of DNA, is that there's a kind of standardized approach. So if someone was choosing new markers, I'm not entirely sure where they would come from, apart from some high-level committees around the world that might be looking at markers that are more discriminating, because one of the problems with databases is that as they get bigger and bigger, there is a higher possibility of two unrelated samples matching, two samples that are truly unrelated appearing to be related. That last comment takes me on to the broader issues which are raised about DNA databases and issues of access, whose samples should be kept, how long should they be kept, why are the samples kept in the first place, and why are they taken from people in the first place. And you've probably heard this on numerous occasions already, and we're only in week 3, the end of week 3. Well, it depends very much where you are in the world. It depends very much what jurisdiction you're in, what legal system you're in. The specific legislation for DNA within the UK, the specific legislation for DNA is different between, for example, Scotland and England and Wales. It's different from all of those. You know, the legislation in the UK is different from legislation in the United States. And there are quite big differences between legislation and practice between inquisitorial legal systems and common law legal systems. So it really comes down to a mixture of quite complex social, ethical, legal and political issues about who goes on a DNA database, how long they go on a database for, and in what circumstances the samples might be removed. So let me just give you, and the purpose, I should perhaps say, in most instances, is to detect, prevent and deter crime. So in the United Kingdom, the primary purpose of all the databases in the individual jurisdictions and the United Kingdom database is that it's for the prevention and detection of crime, and for the most part for no other purpose. Who is kept on the UK database? Essentially people who have been convicted of crime. I'm being a little bit simplistic here, just to illustrate. And for a very wide range of offences, from some people would say not very serious offences, whereas some countries will only sample and retain people for more serious offences. And there is no evidence, as far as I can see, certainly no extensive or systematic evidence that has researched these databases to see which is the most effective in criminal justice terms. They tend to be something of political footballs. There are very strong views about the utility of databases in most countries, and there are also people who detract from that and have very negative comments to make. So the very short answer to who goes on databases and for what reason and for what particular offences, it depends a great deal, and my recommendation would be that wherever you are, you should look the legislation up in your country and see what it means to you. And I think it's back to you, Ben.
The next question is, can we pull the microphone closer, please? I think it's about the end of its tether, so to speak. We'll sit a wee bit closer to it, Jim. There's a question come in online to us just in the last few minutes from John Fairhurst. And it is about a case, but I want to talk about it because I think it's a very legitimate and reasonable question. What's concerning, John, is that this is a case about shooting, and we haven't said anything anywhere about doing gunshot residue examination. The basic answer for that is, what useful purpose would that serve? You have two people who were shot in or around the vicinity of a closed environment like a car, and you find gunshot residues on them. So what? What's it going to tell you? All it's going to tell you is that a gun's been fired in that area. It's not going to tell you whether they fired it, whether someone else fired it. Let's say there's gunshot residues in Mr. Ward's hands. Maybe it's because he got them off his wife where she was shot in the head, or maybe he got them self-protection, trying to fend off the gunman. So the number of instances where the probative value of gunshot residues in this sort of investigation is high is not large at all. So that's why it wasn't done. And just as an aside, I keep referring in responses to you, taking back to the real case. GSR's work were done in that case, and they were an absolute waste of time and money, so we kind of followed that. Moving on to other questions associated with it, Gordon Murphy wants to know why the BPA assumes that the blood droplets are travelling in straight lines. Wouldn't gravity cause them, especially low speed droplets, to travel in an arc? And the way you phrased your question, Gordon, actually answers it as well. They're not travelling slowly. These are high energy impacts. They're going boom. It's like spattering a frying pan in a kitchen. So essentially they're going to travel in straight lines for the short distances that we're talking about. You could also argue, slightly tongue-in-cheek, if you look at vast distances, this is murdered by the law. If you went out to the middle of Loch Lomond on a still, quiet day with no rain, which is not every day, in your boat, it wouldn't move. Because essentially, in that spatial dimension, the world is flat, it's not round at all. The curvature is so slight, it behaves as if it's flat. So I could put my tongue in cheek and say it's because the world is flat. We've also got a question, I think, that Jim and I, in the few minutes we've got left, would like to perhaps backward and forwards a little bit, and that's from Brian Ferrer. And Brian wants to know whether expert witness testimony should be taken as fact or opinion. The theoretical response to that is, by definition, an expert witness is someone who, because of their training, expertise, or knowledge, is allowed to give an opinion in court. Everybody else, a lay witness, is only allowed to talk to fact. And in fact, this has been extended in some decisions about it's not only a matter of the expert witness testimony is allowed to be given as an opinion, it's actually expected that it's an opinion. But this leads to all sorts of things. Where is the boundary between fact and opinion? As we saw recently, going back to the fingerprints weekend in the Shirley McKee case, and the report that came there, where it said, oh, fingerprint experts should understand that their evidence is opinion. And I think you've got a fairly intimate knowledge of that case and decision.
Well, it is opinion, and Bill has set out the kind of legal basis for experts pretty clearly. But it's probably worth distinguishing between this business of facts in this area. It can be quite slippery and quite hard to nail down. But in my view, quite a lot of expert evidence is what I would call technical fact. It is not necessarily information or knowledge that I would expect a court to be aware of or initially understand without the expert's guidance, and certainly I wouldn't expect a jury to be familiar with it. But if you put two experts together, they would see lots of this stuff as fact. The DNA analysis shows a particular set of loci and matches one person and eliminates another person, and so on. So there's quite a lot of scientific or technical fact in there. What's interesting about it, of course, is that for the most part, and again, I'll invite Bill to come in here, when you have opposing experts giving evidence, it is rarely the facts that are challenged. It is rarely that technical fact. What's much more likely to be argued about in court by the prosecution or the defence is what that means and how it's interpreted, and that's why the opinion is so critical. It's not so much what is found, it's what is made of it, either by the expert or by the prosecution in asserting their case, or how the defence counter that if they choose to do so.
Yeah, and I think Jim's last point is very valid. Most of us, most of us, a lot of us are operating in adversarial legal systems, not inquisitorial or inquisitorial related legal systems. And it's really, to a certain extent, it's like a tennis match. And one of my friends, and somebody known to Jim as well, used to say that the oath that you take in court should be changed to swear to tell the truth, the whole truth, and as much of the truth as these blankety-blank lawyers are letting you tell. And it is right, you've got to be right, but it is, there's an art to being an expert witness as well as the expertise side of it. But moving on to some other questions that have come in from Twitter, Anne Mitchell asks, how accurate or useful in solving a crime would a DNA photo fit be? And I think, Anne, I recollect seeing you involved in thread and discussions in this. And this, for those of you who don't know, was probably stimulated by an article, I think it was either yesterday or the day before, in that highly reputable scientific journal, the Daily Mail, which, for those of you who aren't in the UK, is, shall we say, a popular...
Red top.
Red top. It's not exactly the most, well...
Incisive?
Incisive, thank you.
Sophisticated?
Sophisticated of the newspapers. We have a very large following. Because there's been some stuff come out from research that says, as we're getting more and more information, more and more knowledge of what's coming out of the Human Genome Project and follow-ups to it, there are little bits and pieces coming out that give indications about the genetic basis of physical features. So the obvious question is, given that you don't always get an eyewitness who actually saw somebody, and given, as we said in week one, even if you did, there's just so much evidence that questions the ultimate reliability of eyewitness testimony. Wouldn't it really be nice if you could get the objective scientific DNA information related to these genes that are talking about what we look like, and get a photo bit out of it? Yeah, it would. Is it going to happen today? No. Tomorrow? No. Ever? I'm not so sure. And the reason why I'm not so sure about it is when you delve into genetics, I think some folks thought that when they did the Genome Project, what would come out of that would be one gene for absolutely everything. And it's not like that at all. And that's why we put the bit in that module that a lot of people have questioned, do we really need this, and was it in the right place, with some of the background to the Human Genome Project and all the names from it. You know, it's barely more than 20,000 genes there. Half of them, or more than half of them, we share with things like flies. So this whole question of why we don't look like a fruit fly, and why I don't look like Jim, but we're both recognisable as humans, It's enormously complex. There's a lot of genes, and there's a lot of non-directly genetic influences in the sense that they're not related to the coding genes that's present in DNA that determines these sort of person-to-person variations. I think there's too much interaction and too many extraneous factors involved that it's a possibility, but I don't think it's a realistic one.
Here's a thought that I think is useful here when you're both weighing up evidence and trying to get some sense of the extraordinary variation that is present in DNA. And I largely agree with Bill. This interaction between genes is not yet understood, but 99%, it's either 99 or 98, but I think it's 99. 90% of the human genome is shared with the chimpanzee. So the entire amount of variation for the whole human species resides in that 1%. Think about that when you think that something matches 99% and whether the two things are indistinguishable or not. As a kind of closing kind of comments, I think we're coming very close to the end. I'd quite like to go back to one of the comments that Bill made in response to the question which I think came from Enrico, which was about firearms discharge residues, because I don't think we talk about this much in the case for the reasons that Bill has given. But to link the firearms discharge residues to the issues of expert evidence and how important, and facts and interpretations and opinions, I would recommend that those of you who are interested look up, I think it would be called R.V. George. So this is Barry George. This is the man who was arrested and convicted of shooting Jim Dando. It's a very famous crime in the United Kingdom. Barry George was convicted, and as part of that conviction there was a single particle of firearms discharge residues involved. And after 2 appeals, in the second appeal, it was quite clear that that single item of evidence could not be relied on. Now that's a very, very simple summary of the story. The other thing that happened in that case of interest for those of you, I guess in any kind of jurisdiction, but particularly those in adversarial systems, is that part of the prosecution case was that was compelling evidence of George's guilt. And Bill and I have discussed this, and I've discussed it with loads of forensic scientists, and I don't think I've met one yet that agrees that that is significant evidence of anybody's guilt. Not that they would view it in those terms, but they would view it more in the terms of the connection between that individual and a firearm, or the firing of a firearm. But look at that, you'll probably find that online under England and Wales, under England and Wales Court of Appeal, EWCA, and then George. And it's quite a short case, and it is very illuminating in dealing with facts, how they are rendered in prosecutions, the role of the expert witness, and despite all our best efforts, how these things can go really quite badly wrong.
Time for one more? Yeah. Robert Knights asked us, can we explain the prosecutor's fallacy? And he says, I feel this is relevant to contextualise the issues in court when likelihood rates or chances ratios are hurled about. Many thanks. Prosecutor's fallacy is dead simple, and I think it's been unnecessarily complicated and fogged by throwing likelihood ratios into it and all the explanation to say that you see it basically says if we take DNA and we've got a profile that's got a frequency of 1 in a billion, that is not the same as saying that the chances that this person didn't do it are 1 in a billion. These are two entirely different things. You cannot directly take that population frequency information from the evidence and transplant it into any likelihood, I'll use that bad word, any probability of innocence or guilt. It's an entirely different question, and I'm not. There's no causal linkage between the two. Okay.
Thank you, Professor Fraser and Dr. Tilston. To all those watching, we look forward to receiving your feedback. Don't forget you can use Twitter to contact us with any feedback and any comments you may have. Don't forget to use the hashtag FLForensic. We look forward to seeing you at the next Google Hangout. We hope you will enjoy week 4. From us here in Glasgow, thank you and goodbye.